Welcome back to MCB 170, Society and the Brain. This is lecture 23, which corresponds to part 1 of module 12. The title of module 12 is The Aging Brain. In module 12, we'll be discussing the neurobiological, psychological, and sociological aspects of aging, of dementia, of neurodegeneration, and of death. Physician Atul Gawande has had a lot of experience with dying patients. He notes that people care deeply about how the world will be after they die. I think this is an observation of profound importance for the way that people adjust psychologically to death, not only their own death, but the death of their loved ones. We'll discuss that more in part 2. In part 1, I'd like to focus on neurobiological aspects. Normal aging is accompanied by many changes in the brain. The most easily measured are decreases in brain volume. The brain loses both white matter and gray matter with age. Volume decreases are observed in all regions of the forebrain. Shown in the inset are scans of two elderly brains. On the right, a healthy brain, on the left, a degenerating brain. In this particular case, what distinguishes these brains is that the healthy brain comes from an individual who had a high level of vitamin B12, and the brain on the left, an individual with a low level of B12, vitamin B12. In the healthy brain, you can see lots of white matter, the light areas, and gray matter, the darker areas, for example, at the surface of the cortex here. Recall that white matter is composed mainly of axons, myelinated axons, that connect neurons in the cortex and other regions. These darker areas are ventricles. The ventricles are places in the brain that are filled with cerebrospinal fluid. Cerebrospinal fluid is like blood plasma. Blood plasma is the fluid that's left after the red and white blood cells are removed from blood. Normally, the ventricles are rather small. In the degenerating brain, the ventricles seem to expand, filling with fluid, cerebrospinal fluid, kind of taking up the space left behind by dying neurons. In neurodegeneration, neurons die. This volume decrease, neuron death, is especially acute in frontal cortex and hippocampus, leading to age-related impairments in cognition and memory. We generally think of dementia as loss of memory, but it also involves loss of cognitive ability. Changes in brain volume, drastic changes in the structure of the brain, are accompanied by changes in the function of the brain. Normal aging is accompanied by decreases in overall cerebral blood flow and rate of oxygen metabolism, which indirectly indicates regional brain activity. Neurons are dying in the degenerating brain. So overall, the degenerating brain has less neural activity than a healthy brain. But there are also shifts in activity in the older brain. The hemispheric asymmetry reduction in older adults, HAROLD, is an increase in activation symmetry in the frontal lobes. Wait a minute, shouldn't symmetry be a good thing? Isn't symmetry a sign of beauty and health? Symmetry between the frontal lobes is not a good thing. Neither is the posterior-anterior shift, PASA, which is a reduction in occipitotemporal activity coupled with an increase in frontal activity. Wait a minute. Shouldn't an increase in frontal activity be a good thing? Nope, not necessarily. These changes may reflect functional compensation in older adults, in which a decline in normal sensory processing in posterior regions is compensated by increased activity in anterior regions like the frontal lobes. How do we know that these increases in symmetry are bad? Younger brains show PASA during especially challenging cognitive tasks, suggesting that the aging brain has to work harder to accomplish normal tasks. So here's a view of the human cortex that we've shown before. This is the front of the brain and the back of the brain. And so we know that we're looking at the left hemisphere, a side view of the brain looking at the left hemisphere. Of course, the right hemisphere is on the other side in the plane of the screen. Harold is an increase in symmetry in the activity of the frontal lobes on either side of the brain. PASA is kind of an increase in the fore and aft activity of the brain. This is the central sulcus, which separates the frontal lobes from the parietal, occipital, and temporal lobes. In PASA, there's a decreased activity in the back of the brain, which is compensated by an increased activity in the front of the brain, apparently on both sides, left and right. So Harold and PASA increase the symmetry of activity in the brain, left and right and front to back. This increase in the symmetry of activity in the brain, of diffuse activity all over the brain, is essentially a consequence of decreased overall brain activity. The brain is degenerating, parts of the brain are losing their normal function, and overall the brain has to work harder to do things that it did more easily when it was younger and more healthy. The reason why there's more diffuse, more symmetric activity in the aging brain is because the aging brain shows less specificity during task-specific behaviors. In the young brain, increased proficiency on a task is associated with decreased overall activity, but increased activity in specific regions. In the aging brain, this activity change seems to reverse, and activity regresses to the more diffuse pattern. Let me give you an analogy. A good swimmer swims efficiently. A good swimmer only makes the movements that she needs to make. The rest of the body can relax. A bad swimmer flails around in the water, kind of moving everything symmetrically in an uncoordinated way. So the young brain is like a swimmer who can swim efficiently. The old brain is like somebody who's flapping around in the water, not getting anywhere. The more diffuse pattern is a reversal of activity changes. So a young brain, an inexperienced brain, has a lot of overall activity as the young brain tries to figure out how to perform cognitive tasks efficiently. When they learn that, then brain activity changes to specific regions, mediating specific tasks. Other parts of the brain can essentially relax. In the elderly brain, there's neurodegeneration. Those specific regions lose their function. Again, the brain overall has to work harder to accomplish those tasks. This reversal has been attributed to loss of prefrontal activity, which is thought to provide top-down modulation that enhances the processing of relevant input but suppresses the processing of irrelevant input in the younger brain. We discussed this in the module on brain development, where we showed how the development of the prefrontal cortex can help us control ourselves, can suppress irrelevant information, and can suppress unwanted movements. Not only that, but the development of our frontal lobes, and specifically our prefrontal cortex, allows us to control ourselves in general. It helps us to conform our behavior to our moral values and ethical principles. In the aging brain, that ability is lost. So dementia not only involves loss of memory, it also involves loss of self-control. There are several theories concerning the physiological processes underlying brain aging. Here are four. The dopamine hypothesis says that cognitive decline is due to dopamine dysregulation in multiple brain regions, including the striatum and its projections to frontal cortex. The frontal hypothesis says that cognitive decline is due to decline in frontal lobe efficacy. The inhibitory control hypothesis, which says that cognitive decline is due to decreased efficiency of inhibitory processes, which is related to frontal lobe deficiency. and the speed of processing hypothesis, which says the cognitive decline is related to the decrease in speed with which older brains process information. This would be related to loss of myelinated axons in the degenerating brain. It's likely that these four are not separate but highly interrelated processes. That, for example, the inhibitory control hypothesis is related to the frontal lobe hypothesis because the frontal lobes mediate inhibitory control. What about the monoaminergic neurotransmitters? Interesting studies in monkeys have measured the levels of the monoaminergic transmitters in aging. Recall again that the monoaminergic transmitters are dopamine, noradrenaline, and serotonin. It turns out that dopamine levels decrease with age in the monkey brain, but serotonin and noradrenaline stay relatively stable. It's great that serotonin and noradrenaline hang around in the aging brain. It's very sad that dopamine goes down by a lot. Dopamine is really important. It's important not only for movement initiation, but also for keeping us happy. Dopamine is what gives us pleasure, and dopamine is what motivates us to do things. So neurodegeneration, dementia, is associated not only with memory loss and not only with cognitive decline in general, but also with a loss of pleasure in life and lack of motivation. But we generally think of dementia mainly as affecting memory, and it turns out that different kinds of memory are affected more than others. The types of memory that get worse with age include recollection, which involves effortful retrieval of information in the brain. Also accuracy. If you want to retrieve information accurately, it requires healthy cognitive function. Also impaired in aging is source memory, memory for circumstances surrounding a fact, and prospective memory, remembering to carry out future tasks, essentially remembering to remember. I've always been quite bad at that myself. Certain types of memory are relatively unaffected by age. First is recognition, which is automatic recall of learned facts. So once we learn a fact, which can require effort, once that fact is in there, it'll tend to stay even as memory declines overall. Other types of memory relatively unaffected by age are implicit memory, which is memory for habits and acquired skills. So individuals who don't lose language ability, they don't lose their ability to play a musical instrument, for example. Also familiarity, feeling of recognition without details, and flashbulb memory, memory for emotionally salient events. Elderly people would still remember, for example, the weddings of their grandchildren. The pattern of memory loss that selectively affects more effortful memory tasks is consistent with the decline in frontal lobe function in aging, since the frontal lobes are involved in purposeful recollection. Loss of source memory is associated with reduced recruitment of frontal and hippocampal regions. What this is basically saying is that effortful memory, recollection of information, retrieval of information accurately, is a cognitive process, an effortful cognitive process that requires interaction between the frontal lobes and the hippocampus. The frontal lobes and the hippocampus are the two regions of the brain most prone to neurodegeneration. Knowledge for facts, which does not require effortful cognitive processing, actually grows with age as more facts are acquired over the lifetime. And the facts that are acquired aren't trivial facts necessarily. They can be facts associated with life. As an individual lives and observes, that individual will see how, for example, the decisions of certain individuals either benefit or don't a particular individual. They can see through experience how things turn out with people. and over time they can develop wisdom. So even people who have dementia, who have cognitive decline, who have an inability to recall effortfully, still can be wise. But that cognitive decline is pretty terrible. Older adults are much worse than younger adults on visual spatial tasks, which are important for, among other things, driving, at which older adults are notoriously bad. Not all elderly people experience cognitive decline at the same rate. Albert Einstein was still writing books at age 71. Writing books is a highly demanding cognitive task. If you're still writing books at age 71, you're doing pretty well. How was Einstein able to do this? Well, maybe because he had greater cognitive reserves. Cognitive reserves are factors that enhance the resiliency of the brain in accommodating the damage due to aging. It turns out that there are various ways to build our cognitive reserves. We can build cognitive reserves through education and mental exercise. Education overall will build your cognitive reserve. It'll make you more resilient to the damaging effects of neurodegeneration. And so will mental exercise. You may know about certain online brain games, memory games. Those are actually good for your brain. There's some debate as to whether your skill in a particular memory task translates to other kinds of cognitive processes. It may or may not, but you can still improve your ability on certain kinds of like short-term memory tasks through those memory games. The brain, remember, is an organ just like any other organ, and you can increase your cognitive reserves through cardiovascular and general fitness through physical exercise. Diet is also important, especially reduction in caloric intake. We'll be talking about that a lot in this part. Also, increased social interaction is very important. Social interaction requires a lot of cognitive ability. It's kind of a demanding cognitive task. Social interaction kind of is a form of mental exercise. But social interaction can be healthy in another way also, because studies have shown that social interaction, and specifically flirting, can increase the levels of steroid secretion, especially of the sex steroids. Now you might say, in a woman who's postmenopausal, or a man who's post-andropausal, well, their gonads aren't producing very much sex steroids anymore, but their adrenal glands still are. Recall that the adrenal gland produces not only cortisol, but also the sex steroids. So when individuals interact socially, and especially when they flirt, they're going to increase their secretions of steroids, sex steroids and cortisol. And in the short term, that hormone secretion can be health promoting. Finally, we can improve our cognitive reserves through the use of memory cues and mnemonic strategies. Mnemonic is anything that has to do with memory. And what would be an example of a mnemonic strategy? Well, I find as I'm getting older that I often lose my train of thought. But if I focus on the main point before I lead into a digression, I'm more likely to come back to the main point afterwards. And it turns out that these mechanisms that increase our cognitive reserves aren't just a bag of tricks. Education, mental exercise, and sustained mental activity can produce biological changes in the brain that directly help offset the effects of aging. That's fascinating. That's fascinating. Education, physical exercise, mental exercise, social interaction can keep your brain healthy on a biological level. Now for some bad news. You probably know that overeating and lack of exercise increases risk for the most prevalent age-related diseases, including cardiovascular disease, diabetes, and cancers. What you may not know is that overeating and lack of exercise may likewise increase the risk for the most prevalent neurodegenerative diseases, which are Alzheimer's disease, Parkinson's disease, and stroke. Alzheimer's disease, AD, Parkinson's disease, PD, and stroke are the big three neurodegenerative diseases. These are the most common neurodegenerative diseases. Alzheimer's disease results from degeneration of neurons involved in memory, specifically in hippocampus and frontal lobes of cerebral cortex. Parkinson's disease results from degeneration of dopaminergic neurons in substantia nigra that enable movement initiation and continuation. The substantia nigra sits cheek by jowl in the brainstem with the ventral tegmental area. We talk about the ventral tegmental area a lot. The ventral tegmental area produces and secretes dopamine all over the brain. The substantia nigra, which sits right next to it in the midbrain, in the brainstem, also produces dopamine and secretes it more into the basal ganglia, so it's more important for movement control and initiation. Stroke results from occlusion or rupture of cerebral blood vessels, leading to damage of the neurons that receive their oxygen supply from those vessels. These are the big three neurodegenerative diseases. And they're no fun. Here's an elderly gentleman who has Alzheimer's disease. He's using the mnemonic strategy of Post-it notes. He can't remember what's in these different drawers unless he has those. He's also not very happy. He's probably got loss of dopamine, which is making him sad and causing him to have low motivation. Here's an elderly gentleman who has Parkinson's disease. It's difficult for him to move and to initiate movement, so his wife is helping him. Here's a sweet old lady who has a stroke. She's probably lost control because of brain damage due to disruption of cerebral blood flow. In the left side of her body, you can see her, the left side of her mouth drooping down here. She's probably paralyzed in this side of her face. She may be paralyzed for a lot of the left-hand side of her body. It's no fun. These are severe cognitive deficits, and they're accompanied by low dopamine, low motivation, sadness. Here's more bad news for foodies. Epidemiological studies show that low caloric intake is associated with lower risk for Alzheimer's and Parkinson's disease. Conversely, high caloric intake is a major risk factor for stroke. And menopause is not so good either. Menopause is a major risk factor for Alzheimer's disease in women. Andropause in men is not so great either. Maybe social interaction helps offset that a little bit by stimulating the adrenals to produce the steroid hormones. But still, loss of the sex steroids with menopause and andropause is a bad thing in general, and it contributes to neurodegeneration. So what's happening neurobiologically? What's happening on the biological, molecular, and cellular level that causes these neurons to die in neurodegeneration? Well, here are the four main neurobiological causes of neuron death. These are described in an article by Matson. An article by Matson is your reading for the week. Very, very interesting article. I'm going to go through these briefly, but in some detail. I want you to remember these details for your quiz on Friday. Number one, increased oxidative stress can kill your neurons. It involves oxyradical-mediated damage to proteins, lipids, and nucleic acids. What's an oxyradical? An oxyradical is a molecule that contains oxygen, which is highly likely to contribute that oxygen in chemical reactions. An oxyradical is a molecule that will oxidize other molecules. Oxidation is essentially burning. When you burn a piece of paper, you're oxidizing it and destroying it. When metal rusts, it's being oxidized. Oxidation can cause damage to tissues, proteins, lipids, and nucleic acids specifically. Where do oxyradicals come from? Oxyradicals come from several places in your metabolism, in your biology. One of them are the immune cells. When an immune cell wants to attack an invader, it can produce oxyradicals. It can produce oxyradical, oxygen-containing reactive molecules that can kill the invader. Overactivity of your immune system can lead to oxidative stress, which is high levels of oxyradicals, which is damaging not only to invaders, but to your own tissues. It can lead to neuron death. Second, the impaired ability of neurons to regulate ionic homeostasis, resulting in abnormal increases in intracellular calcium. What's ionic homeostasis? As we've spoken about before in the course, all cells have negative ions inside and positive ions outside. They maintain this imbalance in their ionic composition, so that they have more negative ions inside than outside the cell. This is especially important for neurons because neural impulses are waves of positive ionic current, waves of positive ions that enter the axon and are pumped out again. The neural impulses are mediated mainly by the positive ion sodium, but calcium is an even more important positive ion. Calcium can enter cells like neurons, and it mediates lots of functions. We know that calcium entering a synaptic terminal will cause release of transmitter, but calcium also is involved in the regulation of many cellular processes, probably hundreds of cellular processes. If there's too much calcium, those processes become impaired, and that can lead to cell damage and cell death. Third, impaired energy metabolism that may result from or contribute to dysfunction of mitochondria. Mitochondria are the powerhouses of cells. They're involved in the conversion of glucose into ATP. If mitochondria are impaired, neurons can't make energy. They can't metabolize glucose. They can't produce ATP. They'll die of lack of energy. The fourth thing that can kill your neurons is activation of cell signaling cascades that lead to cell death, otherwise known as apoptosis. As you know, you're born with more neurons than you need. Part of normal neural development is death of unneeded neurons. But once your brain develops and you've got your full set of neurons, you don't want any of them to die. When they start to die, you're experiencing neurodegeneration. Cell death or apoptosis involves proteins such as PAR4, the BCL2 family, and the caspase family. These are proteins who mediate molecular interactions, cascades of molecular reactions that can lead to cell death. All of these four mechanisms, cellular and molecular mechanisms, can kill your neurons, and they're not separate. For example, oxidative stress can damage proteins, lipids, and nucleic acids. Calcium channels are proteins. Lipids are membranes. If you damage ion channels and lipid membranes, you can have calcium flowing into cells, which can disrupt their normal functioning. Oxidative stress can also lead to the disruption of mitochondria. And cells who have damage to their mitochondria, who are not maintaining their ionic homeostasis, can undergo apoptosis. And Apoptotic cells can release factors into the extracellular space that can kill other neurons, that can induce other neurons to undergo apoptosis, leading to a more general neurodegeneration. So again, these four mechanisms are not independent, but they can work together and they can gang up on your neurons and cause your neurons to degenerate. Is there anything we can do about that? Well, again, Mattson, an article by Mattson is your reading for the week, subjected mice to an alternate day dietary restriction regime in which they withheld food from the mice every other day. So for a whole day, food was withheld. They could eat the subsequent day, but on the day after that, food was again withheld. This amounted to an approximately 30% reduction in caloric intake, but it extended their lifespans by 30 to 40%. Does this happen in humans? There's very, very strong evidence that caloric restriction increases the lifespan of human beings. It also does in non-human primates. Monkeys who are subjected to caloric restriction will live longer. They'll be healthier to older ages. I think the caloric restriction improvement in lifespan is not as dramatic in primates as it is in rodents, but it works the same way. Mattson found that dietary restriction decreased damage to hippocampal neurons in mouse models of Alzheimer's disease, to substantia nigra neurons in mouse models of Parkinson's disease, and to cortical and striatal neurons in mouse models of stroke. So Mattson has done a lot of work showing how not only caloric restriction, but also exercise and exercising when you're hungry can contribute to the health of your neurons. And how does that happen on the cell and molecular level? Well, Mattson has found that dietary restriction may protect neurons from various forms of cellular damage by basically increasing the production of cells of various proteins that the cells would not produce unless they were subjected to some kind of stress, some kind of mild stress. You subject neurons, you subject the whole animal, including the brain, to a mild stress like caloric restriction, and it causes neurons and other cells to produce proteins they normally would not produce, and those proteins may be protective. Caloric restriction increases the levels of heat shock protein 70, HSP70, and glucose regulated protein 78, GRP78, which can protect neurons against oxidative stress. Caloric restriction also increases levels of neurotrophic factors, especially brain-derived neurotrophic factor, BDNF, which can protect neurons by inducing expression of genes that encode proteins that suppress oxidative stress, such as HSP70 and GRP78, and also stabilize calcium homeostasis. So caloric restriction, which imposes a mild stress, also exercise, and increase the production of certain proteins which are neuroprotective. So a healthy diet is very important, perhaps accompanied by a bit of red wine, not too much. But I'm showing this slide because I want to draw an analogy between wine and caloric restriction. I had the opportunity to visit the Bordeaux region of France. The Bordeaux region is the largest wine producing region in France, and it arguably produces the best wines. So you might think that Bordeaux would have really rich soil, like we have around here. Now, in late October, November, farmers around here are plowing their fields. They've harvested their corn, they've harvested their soybeans. Now they're plowing under the chaff to let it condition over the winter. And they're turning up the soil, they're plowing it. And you can see that the soil in this area is very, very, very dark brown, almost black. It's very, very nutrient-rich soil. You might think that the Bordeaux region of France would have very nutrient-rich soil, but it has the opposite. Take a look at this soil here. It's very gravelly. The French call it la grave or la gravier. It's essentially gravel. It is impoverished soil. Grapes grown here grow under stressful circumstances, and it causes them to produce proteins and other molecules that they wouldn't produce if they were grown in rich soil. This enhances the flavor and the complexity of the wine. So, a little stress is a good thing, just as the best Bordeaux grapes are grown in gravel. For us, dietary restriction and other mild stresses, dietary restriction, vigorous exercise, exposure to cold briefly, uh, exposure to risk, maybe a little bit of social risk, maybe a little bit of adventurous risk, and in turn cause increased expression of neuroprotective proteins. So a little bit of stress can be a good thing. You don't want chronic stress. You don't want chronic stress. Mattson points out that you want to combine dietary restriction and exercise with periods of eating and recovery. You want to alternate stresses with periods of recovery. So mild stresses, like doing exciting, adventurous exercise on an empty stomach, followed by a period of rest and food intake, can greatly improve your overall health and also improve the health of your brain. I think with that, I'll break for lunch. We'll continue with the psychological and sociological aspects of neurodegeneration in part 2.